Welcome to the deep dive. We're here to break down the latest, most complex research and pull out the key knowledge you need, nice and clear. Today, we're jumping into the really challenging world of Crohn's disease. And specifically, we're tackling something that causes a lot of trouble for patients: intestinal strictures.
Yes, this is such a critical area. We've gotten much better at managing the inflammation side of Crohn's disease, the CD, but the scarring that happens afterwards, that intestinal fibrosis, it's still a major, major problem. It's what narrows the bowel, forms these blockages, strictures, and leads to immense suffering, often needing surgery.
And the scale of this problem, clinically speaking, it's pretty big, isn't it? We're not talking about a small number of patients needing operations.
Not at all. The numbers are quite sobering, actually. You're looking at maybe 30% to 50% of kids with Crohn's developing a stricture within just 5 years of diagnosis. And even after surgery, the recurrence rates are frustratingly high. We see them somewhere between, say, 22% and 47%. And here's the real kicker. Despite all the research, decades of it, there are currently zero, absolutely zero, approved anti-fibrotic therapies specifically for CD. That's a huge gap.
Wow, okay, so the current treatments are hitting the inflammation, but they're not stopping the scar tissue from forming. And that scar tissue really dictates the long-term outcome for the patient. Now, this deep dive is particularly interesting because the research we're looking at today points towards something surgeons have actually seen for years during operations, but its exact role was, well, it was kind of a mystery. And that's this thing called creeping fat.
It does sound a bit odd, doesn't it, creeping fat, but it's a very real phenomenon in Crohn's disease. And yes, this new work fundamentally shifts how we should think about that fat. It's not just passive.
Okay, let's unpack this then. Our mission today really is to move beyond just thinking about inflammation inside the bowel wall. We want to zero in on the specific troublemaker cell, understand where it lives, apparently in this creeping fat figure out the molecular on switch for its bad behavior, and get our heads around this idea of outside-in fibrosis, scarring that starts outside the bowel and works its way in.
Right. So first things first, let's define this creeping fat, or CF. It's not just general abdominal fat or weight gain. It's a specific pathological thing where the mesenteric adipose tissue, that's the fatty tissue that normally holds your intestines in place within the abdomen, expands dramatically and wraps itself tightly around the diseased sections of the bowel.
Wraps around it, like actively.
It seems to, yes. It physically encroaches on and covers the inflamed bowel segments. And it's so characteristic of Crohn's disease that surgeons often use it as a visual cue. If they see significant creeping fat during surgery, it strongly suggests CD. And we find it in a huge proportion of strictures, somewhere between 70% and 90%.
70 to 90%. Okay, so it's definitely there, a clear signature. But the big question is it just sitting there like a marker, or is it actually doing something bad? Is it a bystander or part of the problem?
It is absolutely part of the problem. When you look closely histologically, the amount of this CF wrapping correlates directly with how severe the fibrosis is and how thick the bowel wall has become in that area. Surgeons even use the edge of the creeping fat as a landmark sometimes to guide where they need to cut during complex surgeries. But the real breakthrough this research offers is identifying the specific cell type within that fat that's driving the scarring process.
And finding that cell sounds like it was a huge task in itself. The source material mentions a massive effort, pulling together data from loads of different studies.
That's right. They performed a meta-analysis, essentially combining data from multiple human single-cell RNA sequencing studies. This created a large atlas of fibroblasts in Crohn's disease, looking at data from over 400 patients. This large data set allowed them to really pinpoint the specific cell population that stands out as being pro-fibrotic, the ones making the scar tissue.
Okay, the big reveal. What's the cell?
The cell is the CTHRC1 positive fibroblasts.
CTHRC1.
Right. So fibroblasts, in general, are the cells responsible for building and maintaining connective tissue. They make the extracellular matrix, or ECM, think of it like the scaffolding of tissues. In wound healing, this is good. But in chronic diseases like CD, they can go into overdrive and produce too much ECM, which leads to scar tissue or fibrosis. The CTHRC1 positive ones are a specific subtype. CTHRC1 stands for collagen triple helix repeat. Containing one is just a protein marker they express highly. And these particular fibroblasts are uniquely loaded up with gene signatures for making lots of ECM. Their whole program is geared towards building scar.
Like hyperactive construction workers constantly pouring concrete. And critically, where exactly do we find these CTHRC1 plus fibroblasts? Is their location important?
Absolutely crucial. They aren't just scattered randomly. They're found concentrated right at the specific junction where the creeping fat meets the diseased bowel wall. This zone is called the creeping fat bowel wall interface, or the CF-BWIF.
The interface.
Oh,
Okay, so the action is happening right at that meeting point.
precisely. If you examine that interface zone, either visually under a microscope or using computational methods, it stands out. It's clearly the nexus of the fibrotic activity.
You mentioned computational analysis. How do they
Yeah.
measure that activity? How do you sort of quantify the scarring?
They used an interesting computational technique called fibrosis pseudotime. You can think of pseudotime not as real time, but as a calculated trajectory showing how far along a cell is on its path to becoming fully fibrotic, or fully differentiated into a scar-producing cell. So a higher pseudotime value means a cell is further down that path, more mature in its scar-forming state. The analysis showed that cells right at that CF bowel wall interface had significantly higher fibrosis pseudotime values compared to cells in other parts of the strictured tissue, like deeper in the bowel wall or further out in the fat.
Okay, that makes sense. So the interface really is the active frontier, the most advanced point of the scarring process.
Exactly. It confirms this interface is a highly active, highly fibrotic niche.
Right. Now, what triggers these specific CTHRC1 plus fibroblasts? We know they express scar genes, but the research suggests something else makes them special, something about how they sense their surroundings.
That's the other key piece of the puzzle, mechanosensitivity. This is a really important concept. It means these fibroblasts don't just respond to the usual biochemical signals like inflammatory cytokines, although those likely play a role too. They also respond directly to the physical properties of the tissue around them. Things like mechanical tension, stiffness, the composition of the matrix they're sitting in. This physical sensing capability is something that current therapies, which mainly focus on blocking inflammation, completely miss.
Okay, mechanosensitivity. Let's try that analogy we talked about earlier. Imagine the bowel wall, after being inflamed and scarred for a long time, gets stiff and tight, right? Like tightening a guitar string.
Good analogy.
So the CTHRC1 plus fibroblasts are the cells that can actually feel or hear that increased tension. They sense the stiffness. So what's the biological mechanism? How does feeling that tension get translated into making more scar tissue?
The key pathway here is the YA-TAZ signaling pathway. YAP and TAZ are proteins, transcriptional coactivators to be precise. They are part of a larger network called the Hippo pathway, which normally helps control organ size and cell growth. But in this context, mechanical forces like tension or stiffness cause YAP and TAZ to become active. When they're active, they move into the cell's nucleus, and once in the nucleus, they switch on genes that drive the production of more ECM components, more scar material, and also genes that help the fibroblasts move around to migrate. And indeed, the study found high levels of activated YAP, specifically YAP located in the nucleus, which is a sign it's active within these CTHRC1 plus cells, both in the creeping fat itself and right there at the interface.
Okay, so tension leads to YAPTZ activation. YAPTZ activation leads to scar production and cell movement. Seems logical. But you mentioned something earlier about the interface being the hottest spot for fibrosis pseudotime. But where did the researchers actually find the strongest molecular signal of this YAP's pathway being switched on? This is where it gets really interesting and maybe a bit unexpected.
Exactly. It is quite counterintuitive. You'd naturally think the highest yapatase activation signal would be right where the tissue is presumably stiffest and most scarred at the stricture interface itself. But when they actually measured the expression levels of the genes that yapatase turn on genes like CCN1 and SERPIN1, which are downstream targets, those levels were highest not at the interface, but further out within the main body of the creeping fat, away from the bowel wall.
Wait, hang on. The pseudotime scarring measure is highest at the interface where the action is. But the signal telling the cells to scar, the yapatase gene signal, is highest further out in the fat. How does that work?
This is the finding that really supports that outside-in model we mentioned. It strongly suggests that the mechanical stress originating from the tight, stiff stricture is somehow being sensed by these CTHRC1 plus fibroblasts while they are still residing within the mesenteric adipose tissue, the creeping fat, before they even reach the bowel wall. They seem to get primed or pre-activated by this mechanical cue while still relatively far away, like getting their orders before marching to the front line. Then, once activated, they migrate inwards towards the bowel wall, reaching the interface where they then execute the scar-building program.
So they're sensing the tension from a distance, getting activated, and then moving in to do the damage at the interface. It's like an invasion coordinated from afar. How do they navigate and interact with the environment as they move in? What communication pathways are they using?
The analysis showed their main mode of communication or interaction once they get going is heavily focused on the scar itself. They use ECM integrin pathways. Integrins are like little molecular hands on the cell surface that grip onto the extracellular matrix. So these migrating activated fibroblasts are constantly interacting with and likely remodeling the ECM as they move through it towards the interface. They're essentially talking to the matrix, ensuring they can anchor themselves and deposit more scar material effectively. There are also secondary interactions, the data suggests, with immune cells like macrophages, which can release factors like TNF-alpha and TGF-beta, known fibrosis promoters, and perhaps parasites involved in blood vessel formation. But the dominant signal for their activation and migration seems to be this mechanical stress linked through urapatase and then interacting directly with the matrix via integrins.
Okay, quick recap then. We've got these specific cells, the CTHRC1 plus fibroblasts. They are super scar producers. They are mechanosensitive, meaning they respond to physical tension. This response is driven by the Y-apitase pathway. They seem to get activated out in the creeping fat and then migrate to the fat-bowel interface, which is the main site of active fibrosis.
It's a great summary.
So knowing that this mechanical tension seems to activate them while they're still out in the fat, what does that really imply about how they get into the bowel wall? We need proof they actually comes from the fat, right? To confirm this outside-in idea isn't just correlation.
Exactly. Correlation isn't causation. We needed proof of origin. And to get that, the researchers first had to tackle another big challenge, creating a better animal model. Most existing mouse models of inflammatory bowel disease, or IBD, are pretty good at mimicking the inflammation part, but they often don't consistently develop the deep, through-the-wall fibrosis, the transmural fibrosis, and the characteristic creeping fat that you see together in human Crohn's strictures.
So the standard models were missing key features of the human disease they were trying to study. How did they manage to create a model that did mimic the human stricture environment, including the creeping fat?
They smartly focused on that mechanical trigger we've been discussing. They developed a new surgical model in mice. It involves making a small cut, a colotomy, in the mouse's colon. But the key was how they closed the cut. They stitched it transversely, pulling the edges together under maximal tension. By deliberately maximizing the mechanical tension at the repair site, they aimed to really ramp up that mechanosensitive signaling pathway.
Amplifying the signal they suspected was key.
Did it work?
It worked remarkably well. This tension-based surgical model successfully generated both features they needed: long-lasting transmural fibrosis that went deep into the bowel wall and the formation of creeping fat wrapping around that fibrotic area. It was a much better phenocopy of a human CD stricture. And importantly, they confirmed that mice also have TQRT1 plus fibroblasts that behave similarly to the human ones. Okay, perfect. Now they have a model that looks like the human condition. How did they use it to definitively track where those CTHRC1 plus fibroblasts came from? How do you follow a cell's journey?
They used a really elegant technique called localized lineage tracing. It involved using special tiny fatty particles called liposomes, specifically LATMX liposomes. These were engineered to carry a genetic marker. They carefully injected these liposomes directly into the mesentery, the fat tissue of mice that were genetically engineered so that their fibroblasts, specifically those expressing a gene called colony-2, common in fibroblasts, could be permanently tagged or labeled when exposed to the marker from the liposomes.
So like putting a tiny permanent GPS tag only on the fibroblasts originating within that mesenteric fat pad.
Exactly, a very precise local tagging. Then they performed their tension-based surgery to induce the stricture and creeping fat. Weeks later, they could look at the fibrotic bowel wall and see if any of the tagged cells, the ones definitely originating from the mesenteric fat, had migrated into the stricture.
And did they?
They did. The lineage tracing results were clear. They found tagged CTHRC1 plus fibroblast cells carrying the GPS marker from the fat injection clearly present within the fibrotic bowel wall, especially concentrated at that interface zone. This provided direct, definitive proof that these key pro-fibrotic cells originate in the mesenteric adipose tissue, the developing creeping fat, and then actively infiltrate the bowel wall, establishing that highly fibrotic interface. It confirmed the outside-in migration pathway.
That's huge. The fat isn't just reacting, it's the source of these invasive scar-forming cells. This really shifts the perspective. Okay, so the crucial next step, the one listeners really care about. If this pathway is so important, can blocking it actually reduce the scarring? Did they test that?
Yes, and this is where it gets clinically very relevant. They used genetically engineered mice again. This time, they created mice where they could specifically delete or knock out the YAP and TAZ genes, but only in that lineage of fibroblasts originating from the mesentery, the COL1A2 expressing cells we tagged earlier. So they could essentially switch off the YAPTAZ signaling pathway just in the fibroblasts coming from the fat, leaving other cells untouched. Then they performed the tension surgery again.
And the results?
The results were quite dramatic. When YAPTase signaling was blocked specifically in those mat-derived fibroblasts, the mice subjected to the tension surgery showed significantly less creeping fat wrapping around the bowel.
Okay.
A major reduction in the thickness of the bowel wall compared to control mice.
Wow.
And a normalized area of scar tissue measured by trichrome staining, especially in the part of the bowel wall right next to the mesenteric fat. They also saw fewer tagged fibroblasts migrating into the bowel wall.
So let me get this straight. Just by blocking YAPTase in the cells coming from the fat, they were able to significantly reduce the fat wrapping and the scarring in the bowel wall itself.
Yeah.
You didn't even need to target the cells already inside the bowel.
That's precisely the conclusion. The inhibition of YAPTase signaling specifically in the creeping fat-derived fibroblasts was sufficient on its own to significantly ameliorate the intestinal fibrosis in this model.
That is incredibly compelling evidence.
It really is. It strongly suggests that while perhaps initially creeping fat might serve some kind of protective or barrier function, maybe a very primitive attempt at wound containment, the chronic mechanical stress in Crohn's disease flicks a switch. It activates these CTHRC1 plus fibroblasts within the fat via YAPTase, and they then shift from potentially helpful to definitively harmful, driving this destructive fibrosis in an outside-in manner.
So, taking all this together, what does it fundamentally mean for patients with Crohn's disease, especially those dealing with strictures? This seems to rewrite the textbook, doesn't it? It challenges that whole paradigm focused solely on inflammation inside the bowel wall.
It absolutely does. I think it creates a new therapeutic imperative. It suggests that future strategies aiming to prevent or treat fibrosis in Crohn's need to think beyond just the bowel wall itself. They likely need to target both the processes within the bowel and these pro-fibrotic fibroblasts originating from the creeping fat. The success they saw with localized YAPTase inhibition in the mouse model offers a potential therapeutic avenue. Imagine therapies, perhaps delivered locally, that could block YAPTase in the mesentery. This could be crucial for preventing strictures from forming in the first place, or maybe even more importantly, for preventing recurrence after surgery, which we know is a huge problem, especially if surgeons leave some of that creeping fat behind.
It also provides a really neat potential explanation for why strictures are so common at specific locations, like the ileocecal junction, the very end of the small intestine where it meets the large intestine.
That's a great point.
That area is naturally narrower. It handles the transition from liquid to more solid waste, so it experiences high mechanical forces just normally. It's sort of predisposed to high tension, making it a prime spot for this mechanosensitive scarring pathway to get triggered.
Precisely. It fits the model very well. Now, it's important to mention a limitation the authors themselves pointed out. Their surgical model, while great for studying decanescensation, didn't include the chronic relapsing inflammation that's also characteristic of human Crohn's disease.
Right. Reality is probably both happening at once.
Exactly. So future research really needs to explore the crosstalk. How do these mechanical signals interact with the inflammatory signals? How does ongoing inflammation affect YAPTase activation or CTHRC1 plus fibroblast behavior? Understanding that interplay will be key for developing the most effective therapies.
Absolutely. Still, this is an incredible deep dive. We've gone from a massive single-cell data analysis identifying a key cell right through to developing a new surgical model to prove its origin, and finally showing that targeting its activation pathway can actually reduce the scarring. It's fantastic work. Thank you so much for walking us through all that complexity.
My pleasure. It really is a compelling story of how digging into the cellular microenvironment and the physical forces at play can uncover potentially game-changing clinical insights.
Definitely. And before we sign off, we always like to leave you, our listeners, with a final thought to mull over, something sparked by today's discussion. Given the strong evidence that mechanical tension is a primary switch activating the CTHRC1 plus fibroblast scarring pathway, could we envision future management strategies for Crohn's strictures that go beyond drugs? Could there be non-pharmacological approaches? Things designed specifically to reduce mechanical stress at those vulnerable spots in the intestine. Maybe through highly specialized diets or ways to manage gut motility, or even developing physical support devices. Could these work alongside, say, targeted YAPTase inhibitors to offer a multi-pronged attack on stricture formation and recurrence?
That's a fascinating and important question. It certainly opens up a new way of thinking about prevention and management. It highlights how understanding the connection between tissue mechanics, adipose tissue plasticity, how that fat behaves, and the wound healing or fibrosis response could lead to entirely novel interventions down the road. That initial, maybe beneficial CF response getting hijacked by chronic mechanical stress via these specific cells is a powerful concept.